Hello, my name is Lily Scarlett, and for the past 3 weeks I've been at SmtA as a part of the Glaxo Summer Research and Biology experience, where I've been conducting research into the bioremediation of cadmium using cyanobacteria. Cadmium is a highly toxic heavy metal that has been shown to cause organ damage, genetic defects, and cancer, as it is a carcinogen, upon repeated exposure through contaminated food or water. I've been working to modify a protein in cyanobacteria to allow cyanobacteria to effectively remove cadmium pollution from its surrounding water. The type of protein that I'm looking at is called a metallothionein, and metallothioneins exist naturally to help maintain homeostasis by binding to metal ions in the cell surroundings. The gene in the particular species of cyanobacteria that I'm looking at, called Synechococcus elongatus, that controls the production of this protein is called SmtA, and I am working to modify SmtA using site-directed mutagenesis to create a new cadmium binding site along this protein to hopefully improve cyanobacteria's ability to bind to cadmium ions in its surroundings. In the spring, I started culturing my cyanobacteria and growing it in this incubator, and then I extracted DNA so I'd have it ready to work with this summer. I started the summer with that extracted DNA, and I ran a PCR, which allowed me to isolate the particular gene I'm looking at from the entirety of the DNA. Gel electrophoresis then allowed me to verify whether my PCR was successful, and you can see by the bar on the right that I was able to isolate my gene SmtA. I ran another PCR after I collected my DNA back from the gel to help recover the DNA quality. I needed to insert my gene, SmtA, into a vector, which is a circle of DNA that can be inserted into an organism. And in order to do this, I ran what is called a digest, where pieces are cut out of both the vector and the gene that you're inserting to help them fit together. At this stage, I again used gel electrophoresis to verify whether or not my previous step was successful. Here I am inserting the DNA samples with dye, and as the gel runs, the shorter samples of DNA will travel farther along the gel than the longer samples, allowing me to see what length of DNA fragments I have. This is the step that I frequently struggled with throughout SmtA, as my digestion was often unsuccessful and I could not get the clear bars I want. However, you can see in this video, as it looks at the gel, I have nice clear bars, meaning my gel was successful. With my gene and vector successfully digested, I was able to move on to the third step in this diagram, ligation, which is where your insert and your vector are combined into one complete circle of DNA. This piece of DNA can then be inserted into an organism like E. coli to grow and replicate. Here's an image of E. coli that I inserted my vector into. After more trial and error with PCRs, gels, and digests to verify whether or not my transformation into E. coli was successful, at the end of SmtA, I was finally able to work on transforming my gene into cyanobacteria. I would like to thank the Burroughs Wellcome Fund, the NCSSM Foundation, the Glaxo Foundation, Dr. Heather Mallory, and the RBio class of 2024 for making SmtA such a wonderful experience.